Right, welcome back. We're on to the next chapter of control of genetic expression. Right, we have just finished the video on RNA splicing and related aspects, and we're going into the next subject. We just recap to see if anything is left out. We've done the promoter genes, the operator genes, and the regulator genes, and the RNA polymerase, and which control of genetic expression. So I'll just read out the last portion that I think needs a little coverage there. So this was the video that we just watched. So following the video, we have a variation in the longevity of mRNA, provides yet another opportunity for control of gene expression. Prokaryotic mRNA is very short-lived, but eukaryotic transcripts can last hours or sometimes even weeks. And the process of translation offers additional opportunities for regulation by many proteins. For example, the translation hemoglobin mRNA is inhibited unless iron-containing heme is present in the cell. There are also opportunities for post-translational controls of gene expression in eukaryotes. Some translated polypeptides are cut by enzymes into smaller active final products, as illustrated in the figure below, which depicts post-translation processing of the hormone insulin. Insulin is initially translated as a large inactive precursor. A signal sequence is removed from the head of the precursor and a large central portion. The C chain is cut away, leaving two smaller peptide chains, which are then linked to each other by disulfide bridges. The smaller final form is the active form of insulin. Gene expression can also be modified by the breakdown of the proteins that are produced. For example, some of the enzymes involved in cell metabolism are broken down shortly after they're produced. This provides a mechanism for rapidly responding to changing metabolic demands. Gene expression can also be influenced by signals from other cells. There are many examples in which signal molecules example, a hormone from one cell binds to a receptor protein on a target cell and initiates a sequence of biochemical changes, a signal transduction pathway that results in changes within the target cell. These changes can include increased or decreased transcription. The RNA interference system, RNAi, is yet another mechanism by which cells control gene expression by shutting off translation of mRNA. RNAi can also be used to shut down translation of viral proteins when a cell is infected by a virus. The RNAi system also has the potential to be exploited therapeutically. And the RNAi, let me just repeat that portion again for you. Some RNAi virus will invade cells and introduce double-stranded RNA, which will use the cell's machinery to make new copies of viral RNA and viral proteins. The cell's RNA interference system, RNAi, can prevent the viral RNA from replicating. Thus, an enzyme nicknamed dies or chops any double-stranded RNA it finds into pieces that are about 22 nucleotides long. Next protein complex is called RISC, that is RNA-induced silencing complex, bind to the fragments of double-stranded RNA, winds it, and then releases one of the strands while retaining the other. The RISC RNA complex will then bind to any other viral RNA with nucleotide sequences matching those on the RNA attached to the complex. This binding blocks translation of viral proteins, at least partially, if not completely. The RNAi system could potentially be used to develop treatments for defective genes that cause disease. The treatment would involve making a double-stranded RNA from the diseased gene and introducing it to cells to silence the expression of that gene. Right, and the RNA interference system is also explained more completely in the video here. So I'll just play the video for you.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
We're on to the next chapter called epigenetics. Our genome is established when fertilization takes place. And the code remains unchanged throughout our life, except for mutations that may occur in individual cells. Nevertheless, the previous page outlined many internal mechanisms that operate to control the expression of specific genes. In addition, we know that many external forces, external factors, epigenetics, can affect the timing of the gene expression, the degree of expression, and the eventual phenotype that is expressed. These external factors can produce small modifications to DNA, such as addition of metal ions, addition or removal of acyl groups or methyl groups to DNA or to the histones that control the wrapping and packaging of DNA. Attachment of methyl groups appears to reduce transcription or even shut it off. Attachment to acyl groups to histones turns genes on. These biological changes to the genome is known as epigenetic factors, that is, changes occurring above the level of the genome. The methyl group, CH3 acyl group, modification of DNA by the methyl group. In essence, the DNA in our cells provide the code for making functional proteins, and the epigenetic factors act as switches which turn genes off and on. Epigenetic factors are likely to play many important roles, such as turning genes off and on as differentiation proceeds in a growing embryo. As a result, some cells in the pancreas become specialized to produce digestive enzymes, while others synthesize the hormone insulin. Epigenetic factors in utero may have a lasting influence on the phenotype years later. If a pregnant agouti mouse is fed a folate-enriched diet that provides methyl groups, its offspring will have dark coats and be lean and healthy because this particular gene is turned on. If the diet is poor in methyl groups, the offspring will be obese and light coats. In the winter of 1944 to 45, the German army set up a blockade to prevent food and fuel from reaching the western part of Belgium, reducing food consumption to exceptionally low levels for the winter. 50 years later, researchers compared the children who were conceived to starved mothers during that period to mothers who were already in their second or third trimester when the blockade began. They found that the offspring of the starved mothers weighed 14 pounds or more on average, had waist circumferences that were 1.5 inches greater, and were more likely to have developed coronary heart disease. Certain genes are known to predispose to certain types of cancer, such as breast and colon cancer. It is tantalizing to think about the possibility of turning such genes off epigenetically in order to reduce the risk of developing these cancers. Epigenetics and the influence of our genes. This is an 18:40 video from TEDxO by Courtney Griffin that provides an excellent explanation of the interaction among nature, nurture, and epigenetics. So let's watch this video too, right?
Years ago, I found myself in a doctor's office contemplating the nature versus nurture debate from a fresh perspective. You see, I had been trained as a geneticist and had spent my career manipulating DNA and seeing the profound consequences in a lab setting, so I'd always put my money more on the nature or the genetics side of the debate. But as my doctor revealed to me that I was pregnant with identical twins, I realized that my convictions were about to be put to the test. For starters, we had not budgeted on 2 daycare bills at once. So I kind of half-jokingly started to wonder what would be the consequences maybe if we just sent one twin to daycare and maybe just kind of tucked the other one in my office drawer during the workday. Despite their identical DNA, I somehow doubted that things would turn out all that well for the twin in the office drawer. Identical twins have had a profound impact on scientists' understanding of nature and nurture. Studies on identical twins who were separated at birth and raised in separate households have helped us understand different traits that are more affected by nature or DNA versus nurture or the home environment. For example, some traits like IQ or criminal tendencies are more affected by your DNA than the house that you grew up in. On the other hand, other traits like depression in men or your preference for a particular political party are more influenced
by your environment than by your genes. So what about identical twins who are raised in the same home environment? Their nature and their nurture are almost the same. And yet, any parent of identical twins, myself included, can quickly point out differences in their children. One twin may have more of a preference for certain types of foods, or may have more aptitude for a certain sport or a musical instrument. And sometimes, health differences can arise in these children. For example, there are reports of autism or asthma or bipolar disorder arising in one twin at a young age while the other one remains unaffected. How do we explain these differences given that the DNA is the same in these children, and for the large part, their home environment has been the same too? Well, it turns out that some of these differences can be explained by a third, very powerful influence on our lives besides nature and nurture, and this is epigenetics. And I'm going to talk to you today about what epigenetics is and how it impacts your life, even if you're not an identical twin. So before we talk about epigenetics, we need to consider our DNA and how it fits into our cells. Because believe it or not, of the 50 trillion or so cells in your body, each one contains about 6 linear feet of DNA. I mean, if we were to stretch it out, it'd be about as tall as a pretty tall man. So how in the world do we fit that amount of genetic material into something the size of a cell nucleus, which is 400,000 times smaller? Well, the answer is that we do it by wrapping our DNA around clusters of proteins called histones. You can think of histones sort of like molecular spools, and there are about 30 million of these spools in each of your cells. So this helps explain how you fit such a tremendous amount of DNA into a small space. Now we call this combination of histones and DNA chromatin. And while chromatin solves this tremendous packaging problem that the cell has, it also presents a new one for the cell. And this is one of DNA accessibility. Because keep in mind that the functional units of DNA are actually the genes encoded in it. These are the instructions for the cell. These are what tell the cell what to do and who to become. And yet when these genes are tightly compacted into a chromatin structure, the cell is unable to read them. They might as well not even be there. And this is where epigenetics comes in. So epi, meaning on top of, and genetics, your genes, literally refers to a set of instructions that sits down on top of our DNA and our histones. Epigenetic marks are small chemical tags which sit down on our chromatin and can help instruct it whether to compact or decompact. And those instructions can then affect how the cell reads the underlying genes encoded in the DNA. So to show this schematically, some epigenetic marks, shown here in red, can help condense chromatin. And when they do this, they obscure the underlying genes, preventing the cell from being able to read them. They turn those genes off. Other epigenetic marks, shown here in green, can help decondense the chromatin. And when they do this, the gene becomes accessible to the cell. The cell is able to read it and turn it on. Now, these types of epigenetic marks are profoundly influential to our biology. Consider, for example, what is it that makes our cells different from one another? What makes them look and behave differently? What is it that makes a muscle cell, for instance, look different from a neuron? After all, these cells contain exactly the same DNA, but it's their epigenetic instructions that help tell them which genes to turn on and which ones to turn off. And with those different genes at play, these can become very different cells. So you might be wondering, when does all this epigenetic information get laid down on our chromatin? And the answer is that much of it happens during our embryonic development. So interestingly, when you were first conceived and you were just comprised of a few undifferentiated embryonic stem cells, which had the potential to become any cell in your body, your chromatin didn't have many epigenetic marks on it. It was only as your cells began to divide and receive signals and information from surrounding cells that the epigenetic marks began to accumulate, and then the genes began to get turned off and turned on, and the muscle cell became very different from the neuron. This brings me to a really important point about epigenetics, and that is that epigenetic marks can be influenced by the environment. And when I say environment, I don't just mean those surrounding cells that tell a neuron to become a neuron. I also mean the environment outside of the developing embryo. So the food that mom eats, or the prenatal vitamins that she takes, or the cigarettes that she smokes, or the stresses that she encounters at home or at work, can all be transmitted as chemical signals through her bloodstream to her developing fetus, where they can get laid down as epigenetic marks that affect the fetus's own genes and long-term health. Now, this has been shown experimentally in mice. Mice contain a gene called agouti, which makes them obese and yellow and susceptible to diseases like cancer and diabetes. This gene and these traits can be passed down from generation to generation through DNA, so that an agouti mother will give rise to a fat, yellow, disease-susceptible offspring if that offspring contains the agouti gene. Now, here's something interesting about the agouti gene. It can be turned off if silencing epigenetic marks accumulate around it. So, if a pregnant agouti mother is fed a diet which is supplemented with these silencing epigenetic marks, those marks will be chemically transmitted to the DNA of her embryo, where they'll accumulate around that agouti gene and effectively turn it off. Her embryo will maintain those marks, and so it will be born and grow up to be an adult mouse that's thin and brown and healthy. Even though this mother is genetically identical at the DNA level to both sets of these offspring, you can see that the diet that she consumed during her pregnancy can affect the health and appearance of her offspring. Now, this has, of course, implications beyond the mouse world, because studies in humans have shown that women who don't eat well during their pregnancy, who eat bad foods, will go on to have children who are more susceptible to developing obesity and cardiovascular disease. Likewise, if women smoke during their pregnancy, their children will grow up to have a greater chance of developing asthma. These correlations between maternal behavior during pregnancy and the long-term health consequences for their offspring are thought to be linked by epigenetics, much as you've seen here in the case of mice. Now, another important point to make about epigenetics is that these types of marks can be transmitted not only from a pregnant female to her fetus, but also from generation to generation if the marks are put down on our sperm or eggs. So if you're in the audience and you're not pregnant and you're not even thinking about conceiving, think about this, because the lifestyle decisions that you make today can still affect future generations. For example, a long-term study was conducted in Sweden and England that showed that young boys who overate or started smoking during their prepubescent years, as their sperm were starting to develop, went on to have sons and grandsons with significantly shorter lifespans. It's believed that the epigenetic marks that were transmitted by their diet and smoking decisions affected the long-term health of their future generations. This type of epigenetic information, of course, can also be passed through females to their daughters and granddaughters if the epigenetic marks are laid down on their eggs. Now, this idea of transgenerational inheritance of epigenetic marks is still being debated and studied in terms of humans, but I should add that non-human organisms, mice, flies, worms, there's mounting evidence that this theory holds true. In fact, it's being shown in the lab that over tens of generations, epigenetic marks can be passed down. Now, another thing to know about epigenetics is that they don't just affect us when we're a developing embryo or when the sperm and egg that conceived us were developing. They can also affect us after our birth. And this is particularly relevant as we think about our brains, which continue to grow and develop throughout our lives. Take this example from rats. Rats contain a gene called the glucocorticoid receptor, and this gene can be expressed or read in a certain region of the rat's brain, and when it is, it helps the rat cope with stressful situations. So the more receptor that a rat has in this region of the brain, the better it will handle stress. Now, there are studies that have shown that interactions between a rat mother and her pups during the first week of their life can have long-term consequences for how much glucocorticoid receptor those pups will grow up to have in their brains, and therefore how well they'll handle stress. This is how this works. When rat pups are born, their glucocorticoid receptor gene is surrounded by a number of these silencing epigenetic marks. This effectively turns the gene off. And yet, if a rat mother extensively licks and grooms and her pups, basically takes good care of them during the first week of their life, those epigenetic silencing marks can be removed from the gene. This allows the glucocorticoid receptor gene to turn back on, and it stays on in those pups' brains throughout their lives, so they grow up to be well-adjusted animals who deal well with stress. If a rat mother ignores her pups, that glucocorticoid receptor gene will maintain those silencing epigenetic marks. They won't go away, and they'll stay in those pups' brains throughout their lives. These rats will grow up to be very anxious in stressful situations. This actually brings up a really encouraging point about epigenetics, in that epigenetic marks are reversible. So if you've been sitting in the audience cursing your parents and your grandparents for their poor lifestyle decisions or for the lack of licking and grooming that you received as a baby, take heart. Because scientists are making terrific progress in designing drugs that can reverse toxic epigenetic marks to help combat certain diseases. This is especially looking promising in the case of certain cancers, which happen to be affected or turned on by aberrant epigenetic marks. This is how this can work. Our bodies have certain genes in them called tumor suppressor genes. The job of these genes is to protect cells from becoming cancerous. But if too many silencing epigenetic marks start to accumulate around these genes, the genes get turned off and they can no longer perform their job of protecting the cell. So scientists have developed drugs which have undergone FDA approval and are in a clinical setting, which can target these silencing marks, effectively removing them from the tumor suppressor genes and allowing these genes to go back to their job of protecting the cell. Now think about it. This is a radical departure from traditional cancer therapy. Historically, we've always been focused on killing cancer cells. This, however, is taking the approach of restoring cells to their original nature, reminding them of what they're supposed to do. This type of therapeutic approach is showing great promise in terms of other diseases as well besides cancer, diseases that are also similarly affected by aberrant epigenetic marks, like diabetes and lupus and asthma, and certain neurological disorders like Huntington's and Alzheimer's diseases. I'm optimistic that this type of therapy is going to hold great promise for our health in upcoming years, but I should caution that one of the challenges as we go forward is figuring out how to target these drugs toward toxic epigenetic marks while leaving alone the beneficial ones that help maintain our health. I want to conclude by emphasizing that there are things that we can do now to positively influence our epigenome. It's not too late. It's not too late to start eating healthier foods that we already know are good for us, like leafy vegetables, whole grains, avoiding cigarettes, cocaine, stress, all of which have been shown experimentally to impact our epigenomes negatively. These are things that you can do to impact your genes and your long-term health. And if that's not incentive enough, they can also impact the health of your future children and grandchildren. I think this concept that we can positively impact our genes is really profound and empowering, because we'd always worked under the assumption that our genes are set in stone, that they're beyond our influence. I want to end today by challenging you and myself to take the opportunity that we have before us to positively impact our long-term health by treating our epigenome kindly through healthy lifestyle decisions. Thank you.
So, in summary, all of our traits and characteristics, our phenotype, are the results of an interaction between our genome, all the genes we inherit, and environmental factors, some environmental factors including our diet, our behaviors, and a myriad of environmental factors including our diet, our behaviors, and a myriad of environmental exposures, influence our phenotype through non-genetic mechanism. For example, one might have a number of genes that predispose an individual to being lean. However, such an individual might still become overweight or obese despite their lean genes as a result, of chronically overeating. Yet other epigenetic factors from the environment can modify the genome in subtle ways without actually changing the code. There are some additional questions. Did you know pink hydrangeas can be made to turn blue by adding aluminum sulfate to the soil? So, the next page. We're into reproduction. Binary fission in prokaryotes. Prokaryotes reproduce by the relatively simple process of binary fission. The single chromosome replicates and each chromosome attaches to a different location on the cell membrane. The cell membrane then begins to invaginate and eventually separates into two genetically identical bacteria. A similar process is used to replicate mitochondria within eukaryotic cells, but the overall process of cell replication in eukaryotes is more complicated. See below. Mitosis. You have the daughter cells and the chromatin condensation, rib formation, or nuclear envelope cytokinesis. Many of the portions have been taught before, but I'm just repeating it for those who are new to the lectures and those who might have missed out some points. Please take screenshots and make necessary notes that could be helpful to your learning process to set the various qualifiers right. Mitosis is the process by which eukaryotic cells replicate by dividing into two genetically identical cells. It is the process by which new cells are formed in the growing embryo and after birth, and mitosis also replaces cells that have died or been shed. In humans, some cells retain the capacity to divide throughout life. These stem cells divide by mitosis and produce daughter cells, which then differentiate into a particular cell type. This provides a way of replacing cells such as skin cells, the epithelial cells that line the respiratory, digestive, and urogenital tracts, and blood cells. Benign and malignant tumors also grow through mitosis. The cell cycle. Cells normally follow a carefully controlled cell cycle depicted below. Many of our cells are mature functioning cells that are not actively dividing. These are cells in the GO phase. This is sometimes called resting phase, but these cells are actively functioning, and they are resting only in the sense that they aren't replicating. The phases in dividing our cells are as follows: G1, when the cell grows in size in preparation for division. S, when synthesis of new DNA replication takes place. G2, when there is a continued cell growth. M, which stands for mitosis, that is when the cell actually divides into two identical cells. The cell cycle is normally carefully controlled by a number of biochemical mechanisms. Laws of control mechanisms can result in abnormal cell division and progression to tumor formation. This is discussed in detail in the online module on cancer. Right, so why don't we just go to that online module on cancer? So sorry, that's not available. So we'll go back. Meiosis. When we get a future chapter on cancer, of course, there would be detailed studies on that once we finish this, because I'll take a separate lecture on that. Excuse me, meiosis. Meiosis is the specialized process by which gametes, sperms, and eggs are produced for sexual reproduction in the ovaries and testes. Recall that humans have 22 pairs of homologous chromosomes and one pair of sex chromosomes. One member of each pair came from the mother, the other from father. The 46 chromosomes are referred to as the diploid 2 in number because there are 2 of each. In order for the fertilized egg to end up with the correct diploid number, sperm cells and eggs must be produced such that each has one chromosome from each pair. In other words, gametes have only 23 chromosomes referred to as the haploid number, that is 1n. Meiosis then is the process which specialized diploid stem cells in the ovary, oogonia, and testis, spermatogonia, produce eggs and sperm which have a haploid number of chromosomes. Thus, each gamete has 23 chromosomes, one from each of the 22 homologous pairs plus one sex chromosome. Now, the video below shows the difference between mitosis and meiosis. Please watch.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
Right, and continuing from there, meiosis shuffles the deck. Meiosis produces sperms and eggs with novel mixtures of the original parental chromosomes due to random assortment separation of homologous pairs of maternal and paternal chromosomes. Results in each gamete randomly getting some maternal chromosomes out and some paternal. Random assortment of 23 pairs of chromosomes can produce greater than 8 million possible combinations, crossing over. After maternal and paternal chromosomes matching up as homologous pairs, they exchange sections of DNA. This further shuttles the genetic deck. So one pair of chromosomes of 23, meiosis 1, meiosis 2. And we go to the next page. Fertilization, oogenesis, and spermatogenesis. Oogenesis, this figure, please take screenshots of these. Right, you need them. That's spermatogenesis, you need screenshots of these. Right, the diagram below summarizes the process of meiosis, fertilization, mitosis. Note that the numbers indicate either the diploid number of chromosomes in cells, 46, or the haploid number 23 in gametes. Right, we go to the next page. Mendelian patterns of inheritance. Gregor Mendel was an Austrian monk who formulated some of the fundamental principles regarding the inheritance of traits. And between 1856 and 1863, he performed thousands of experiments in which he crossbred pea plants with dichotomous characteristics such as color, example yellow or green. Several characteristics Several conclusions were drawn from his studies. The hereditary determinants are finite factors called genes. Each parent has a general gene pair in each cell of for each trait of study. One crosses two pure lines, one which is homozygous for the dominant trait, and that one that is homozygous for the recessive trait. The progeny will be heterozygous, and one dominant allele and one recessive allele. One member of a given gene pair segregates into a gamete, and each gamete carries only one allele for each gene. During fertilization, gametes unite randomly, independent of the genes. We continue with the Mendelian patterns of inheritance. We were at the line number 3. One member of a given gene pair segregates into a gamete, and each gamete carries only one allele for each gene. During fertilization, gametes unite randomly, independent are the genes. The gene that determines whether multiple lipomers will form, referred to on page 6, illustrates a Mendelian pattern of inheritance. A certain gene has two alleles, one that causes multiple lipomers, allele in orange, and one that does not. The dichotomous trait like this one can give predictions about the proportions of offspring by using a Punnett square, which shows the 4 possible pairs of alleles that can occur in the offspring. In scenario number 1 above, the Punnett square demonstrates that only heterozygous gene pairs are possible, so all of the offspring will have multiple lipomers since the lipoma allele is dominant. This is the Punnett square. Mother's alleles, father's alleles, and you check. All of the children will be heterozygous L1 and have the dominant trait. Table 2, Punnett square for offspring of a heterozygous L1 mother and homozygous recessive 2 father. In this case, half of the offsprings on average will be heterozygous and have multiple lipomers, and the other half will be homozygous recessive and be free of lipomers. Punnett square for the gender. And now dominant versus recessive inheritance patterns. Some diseases are inherited, and the pattern of appearance within a family tree would depend on whether the family faulty allele is dominant or recessive compared to the normal allele. For example, the allele for Huntington's disease is dominant. If a heterozygous HH man with Huntington's disease is dominant, and the normal woman HH have children, some of them, about half on average, will have the disease. Individuals shown with a dominant allele like this, the disease occurs fairly frequently in the family tree. In contrast to Huntington's disease, the cystic fibrosis is caused by a recessive allele, meaning that individuals who are heterozygous for the cystic fibrosis allele, shown as CC below, will not manifest any signs or symptoms of cystic fibrosis. As a result, the cystic fibrosis allele can be passed along a family tree with only sporadic appearance of individuals who have signs and symptoms of cystic fibrosis because they are homozygous for the recessive allele. Huntington's disease. Take a screenshot of this, it's easy to understand and make your inferences. The Galtonian patterns of inheritance. Mendel's studies focused on dichotomous traits in plants, such as the color of peas, green or yellow, and plant size, tall or dwarf, but many traits have continuous distributions. Right? Such as height, weight, and intelligence. Galton was a contemporary of Mendel's who studied the intelligence inheritance of continuous characteristics. The idea that characteristics might be blended or averaged occurred to him when he noted that very tall fathers tended to have sons shorter than themselves, and extremely short fathers tended to have sons taller than themselves. He referred to this as a regression to mediocrity, and he concluded that height doesn't follow the inheritance patterns of the dichotomous trait that Mendel studied, and that the phenomenon of dominance didn't apply here. Mendelian inheritance patterns predicted some disease, but only a few, and Galtonian genetics was limited by the inability to predict outcomes. R. F. Fisher, a British statistician and an evolutionary biologist, was able to reconcile these two patterns of inheritance by showing that the inheritance of quantitative traits can be reduced to Mendelian inheritance if multiple genes are involved. For example, suppose the average height in a population is 68 and height is determined by one gene with possible 3 alleles, the HO, which neither adds nor subtracts from the average, the H plus 2, which adds 2 to height, and H minus 2, which subtracts 2 from height. Suppose all the HO alleles is twice as common as the other 2 alleles in the population. If these are codominant alleles, a Punnett square would predict the following inheritance patterns: distribution of heights. Now, if instead just one gene, there were 2 genes that determine height, both of them had 3 possible alleles, as described above, then a Punnett square would predict a distribution with more categories and finer differences among the categories. It is easy to imagine that if there were 3 or more genes that were also determinants of height, the distribution would increasingly conform to a Gaussian distribution. In fact, a similar model with 3 gene loci, each with 3 alleles, looks very much like a bell-shaped distribution. As a result, continuously distributed characteristics are likely to be determined by a finite number of genes which have codominant alleles. And once again, it is important to point out that many environmental factors are likely to interact with the genotype to produce the final phenotype. Sex-linked inheritance. Earlier in this model, it was noted that X and Y chromosomes are physically different from one another, and the Y chromosome is much shorter, and the Y chromosome only has about 9 gene loci that match those on the X chromosome. As a result, almost all the alleles on a male single X chromosome are expressed, since there is no alternate dominant allele to mask them. This results in a distinct inheritance pattern for traits that are encoded on the X chromosome. For example, there are many types of color blindness. All of these conditions are inherited, and most of them are sex-linked because they are caused by defective alleles carried on the X chromosome. Red-green color blindness is fairly common, mild form of color blindness, which can be found in about 6% of the male population. It's far less common in females. This form of color blindness is caused by a recessive allele, and the inheritance pattern is illustrated in the figure below. The defective allele is only carried on the X chromosome, as is the normal allele, which is dominant. A heterozygous female, as shown above, would have normal color vision, but she would be a carrier of the allele for red-green color blindness. Now, suppose that she marries a man with a normal red-green color vision. He only has the C alleles, and they have children. On average, half of the offspring will inherit mom's defective X chromosome, and half will inherit the X chromosome with the normal allele. The dad can contribute either an X chromosome with a normal allele or a Y chromosome with no allele for color vision. The Punnett square illustrates the possible combinations of alleles that will occur in the offspring. As can be seen, and as you can see, there are 4 possible results. Both males and females can inherit the allele for color blindness, but will not be expressed in any other females because the normal alleles is dominant, so the heterozygous CC females, will be carriers of the trait. But a normal color vision. In contrast, the males who inherit the defective allele will be colorblind because the Y chromosome doesn't have an allele to oppose it. The next topic will be the evolution of antimicrobial resistance. Right? The term antimicrobial is a general term that encompasses drugs, chemical or other substances, and that either kill or slow the growth microbes. These include antibacterial drugs, antibiotics, which will kill bacteria, antiviral agents, which will kill viruses, antifungal agents, which will kill fungi, antiparasitic drugs, which kill protozoa. Scottish scientist Alexander Fleming is widely credited with the discovery of the antibiotic properties of penicillin in 1928. Although an earlier report that had noted the ability of penicillin mold to kill bacteria as early as 1897. There are scattered reports of penicillin mold being used to treat gonococcal infections of the eye of newborns as early as 1930, to treat gonococcal infections of the eye of newborns as early as 1930, but it wasn't until the 40s that penicillin began to be used as clinical infections. It was soon recognized to be a truly remarkable drug. Small doses cured infections caused by Staphylococcus, Streptococcus, Neisseria, syphilis, and many other bacteria. Penicillin use became increasingly widespread and indiscriminate, and resistant strains of bacteria emerged. Increasingly large doses of penicillin were required to cure infections, and some strains were entirely resistant. Other antibiotics were developed, and synthetic penicillin-like drugs were introduced, but strains of bacteria resistant to these new antibiotics also emerged. At first, these problems were dismissed, but by the 1980s, it had become clear that antibiotic resistance was an important and growing problem. The 1950s studies were published showing that animals were given low doses of antibiotics, gained more weight more rapidly, and practice of including antibiotics in grain to promote the growth of cattle, poultry, and swine became widespread, further compounding the problem of bacterial resistance to antibiotics. Antibiotics kill or inhibit the growth of susceptible bacteria. Sometimes one of the bacteria survives because it has the ability to neutralize or evade the effect of the antibiotic. One bacteria can then multiply and replace all the bacteria that were killed off. Exposure to antibiotics therefore provides selective pressure, which makes the surviving bacteria more likely to be resistant. In addition, bacteria that were at one time susceptible to antibiotic can acquire resistance through mutation of their genetic material or by acquiring pieces of DNA that code for the resistance properties from other bacteria. The DNA that codes for resistance can be grouped in a single, easily transferred package. This means that bacteria can become resistant to many antimicrobial agents because of the transfer of one piece of DNA. Penicillin-resistant bacteria. Penicillin's ability to kill bacteria was due to its ability to inhibit the bacterial enzyme that was essential for synthesis of the bacterial cell, or penicillin's ability to do this depended on a key structure called a beta-lactam ring. Resistance to penicillin initially occurred as a result of mutation in a bacterium that created an enzyme penicillinase, which was capable of breaking down the beta-lactam ring. Bacteria that possessed this resistance first evolved in hospitals, but they rapidly spread to the wider community at large. The gene encoding for penicillinase resided not on the bacterial chromosome, but on an extra circular ring of DNA referred to as plasmid. This extra piece of bacterial DNA can be replicated and transferred from a resistant bacterium to one that was previously susceptible by a process referred to as bacterial conjugation. Methicillin-resistant Staphylococcus aureus, mRNA. Methicillin, a chemically modified version of penicillin, was introduced in 1959 to treat infections caused by bacterium resistant to penicillin, but it was effective against a narrow spectrum of bacteria. In addition, strains of Staphylococcus resistant to methicillin was reported as early as 1961. These strains had acquired a gene MECA, that's MECA, which inactivates methicillin by encoding for a protein that binds to it. The MECA gene is carried on an extra-mobile genetic element, the staphylococcal cassette chromosome, SEC-MEC. Initially, mRNA strains were encountered early in hospitals, but in the late 1990s, mRNA was found in the community at large and quickly spread worldwide. Students in WHO has created a 7-minute public service announcement that explains how mRNA evolved in hospitals and eventually escaped to the community to cause cases of so-called community-acquired mRNA. Bacterial acquisition and antibiotic resistant genes. As we noted, novel genes arise from random mutations, and occasionally such a mutation may confer a bacterium with resistance to an antibiotic.